We propose that all Bush restrictions on embryonic stem cell research be lifted. Embryonic stem cells are stem cells derived from the inner cell mass of an early stage embryo known as a blastocyst. Human embryos reach this stage 4 to 5 days post-fertilization, at which time they consist of 50 to 150 cells. Embryonic stem cells are pluripotent. This means that they are able to differentiate into each of more than the 220 cell types in the body. Due to their plasticity and potentially unlimited capacity for self-renewal, embryonic stem cell therapies have been proposed for regenerative medicine and tissue replacement after injury or disease. Currently, researchers receiving federal funding do not have access to the stem cell lines they need to conduct their research thoroughly, and forces researchers to use alternative approaches of science. Embryonic stem cells offer a wide range of research possibilities. According to the National Institute of Health, studying stem cells will help us understand how they transform into the dazzling array of specialized cells that make us what we are. Some of the most serious medical conditions, such as cancer and birth defects, are due to problems that occur somewhere in this process. A better understanding of normal cell development will allow us to understand and perhaps correct the errors that cause these medical conditions. The nation's leading scientists believe that embryonic stem cell research has greater potential than research of adult stem cells. Embryonic stem cells are extremely important because they can grow indefinitely in a lab and let scientists study what happens in the first stages of cell development. Only embryonic stem cells offer this potential. The limited number of stem cell lines available for federal funding under the Bush restrictions limit research breakthroughs and slow research efforts. According to the National Institute of Health, there are currently only 21 stem cell lines available to researchers using federal funding. These lines were those that were created before August 9, 2001, when Bush implemented an executive order limiting federal funding for stem cell research only to the embryonic stem cell lines that already existed. This was a gesture to his conservative Christian supporters who regard embryonic stem cell research as destroying a potential life. Some methods of extracting stem cells do destroy the embryo, but new ways of extracting the stem cells leave the embryo intact. Senator Orrin Hatch, a Republican, is the only member of Congress who opposes abortion rights, but publicly supports the use of human embryos, including cloned embryos for research. The embryos used in research will be discarded either way, so it's better to research them than to throw them away. According to the California Institute for Regenerative Medicine, the in vitro fertilization embryos may be leftovers after a couple has completed their family. Other embryos might be ones the couple chose not to implant due to genetic mutations. Still, other embryos might be malformed in some way that causes them to be rejected for implantation into the mother. Thousands of these embryos are discarded every year. These need to be studied instead of thrown away. Many scientists agree that the new lines need to be studied under the Bush restrictions, and they cannot be federally funded now. John Gearhart, director of the Institute for Regenerative Medicine at University of Pennsylvania in Philadelphia, says, "There are hundreds of embryonic stem cell lines out there that have been made under the best conditions, and some of them are patient-ready. They have greater utility, performance, and safety than the Bush-approved lines." Gene Loring, director of the Center for Regenerative Medicine at the Scripps Research Institute in La Jolla, says, "Using the old lines is like being required to use Windows 98. The current policy also prevents federal funds from being used to work on dozens of diseases because many disease-specific lines were created after the deadline of August 9, 2001. None of the 21 stem cell lines approved for use by the National Institute of Health carry a gene defect for many genetic diseases. Therefore, restricting federal funding to only those lines hinders progress in studying these afflictions and delays treatment to millions of Americans. In the years following the restrictions of approved cell lines, many advancements were made, but we can no longer build upon those advancements with the 21 approved lines. It is time to expand federal policy. The Bush restrictions must be lifted, and researchers receiving federal funding must be allowed to access all stem cell lines. Lifting the restrictions would allow research to be performed on all types of cells using federal funds. This would allow researchers to make better progress in their quest to cure disease.